The vast majority of caloric restriction research in humans has been conducted in people who are overweight or obese. The interventions in those studies produce benefits, but it is difficult to know how much of the benefit comes from weight loss itself versus the caloric restriction. The CALERIE trial, which stands for Comprehensive Assessment of Long-Term Effects of Reducing Intake of Energy, addressed a different question: what happens when healthy, non-obese adults reduce their caloric intake by a sustained amount over 2 years? The Phase 2 trial, published in the New England Journal of Medicine in 2015, enrolled 218 adults at 3 U.S. research centers. Participants randomized to the caloric restriction arm achieved approximately a 12% reduction in caloric intake over 2 years. The cardiometabolic outcomes across that 2-year period are what this video examines. In this video, the CALERIE trial design and what made it methodologically distinct, what the 12% caloric restriction achieved in terms of weight loss and metabolic outcomes, what it found regarding bone density and quality of life, 2 concerns often raised about sustained caloric restriction, the limitations of the evidence, and 3 takeaways for adults over 55 thinking about energy intake. Quick note before we continue: this is educational content based on published research, not medical advice. Talk to your physician before changing your regimen. Now, the study. Why does caloric restriction interest longevity researchers in the first place? The evidence from animal models is extensive. Reducing caloric intake by 20 to 40% without causing malnutrition extends lifespan in organisms ranging from yeast to roundworms to fruit flies to rodents, often dramatically. In rhesus monkeys, 2 long-running studies from the National Institute on Aging and the University of Wisconsin produced somewhat inconsistent results, but the aggregate data suggest that caloric restriction in primates delays the onset of age-associated diseases. The biological mechanisms proposed include reduced oxidative stress from lower metabolic rate, activation of AMPK and sirtuins pathways, reduced insulin and IGF-1 signaling, and reduced core body temperature. Whether these mechanisms translate meaningfully to the human lifespan in a world where caloric restriction is voluntary and difficult to sustain is the core empirical question the CALERIE trial began to address. The CALERIE Phase 2 design. 218 healthy, non-obese adults aged 21 to 51 were enrolled at 3 sites: Pennington Biomedical Research Center, Tufts University, and Washington University. Participants were randomized 2-to-1 to caloric restriction or an ad libitum control group. The caloric restriction arm targeted a 25% reduction in energy intake from baseline. Participants received intensive dietary counseling, food provision assistance, and behavioral intervention to support adherence. The intervention lasted 24 months. Outcomes measured included body weight, cardiometabolic biomarkers, body composition via DXA scanning, bone mineral density, quality of life, mood, and sexual function. This breadth of outcome assessment was designed specifically to capture potential adverse effects alongside any benefits. What level of restriction was actually achieved? The target was 25% reduction. The achieved reduction, measured through doubly labeled water, the gold standard method for energy expenditure measurement, was approximately 12% over the 2-year period. This gap between target and achieved restriction reflects the difficulty of sustained voluntary caloric restriction in free-living adults, even with intensive support. The actual restriction achieved was approximately 279 fewer calories per day than the control group. Body weight in the caloric restriction group decreased by an average of 7.5 kilograms at 12 months and was maintained at approximately 7 kilograms below baseline at 24 months. In the control group, weight was essentially stable. The cardiometabolic outcomes at 24 months. Compared to the control group, the caloric restriction group showed statistically significant improvements in LDL cholesterol, triglycerides, blood pressure, insulin sensitivity, and markers of metabolic syndrome. C-reactive protein, the inflammatory marker, was also significantly lower in the caloric restriction group at 24 months. The composite cardiometabolic risk score showed meaningful improvement relative to controls. These findings are consistent with the weight loss achieved, which is not surprising. The specific contribution of caloric restriction per se, independent of the weight lost, is difficult to disentangle in a human free-living trial. 2 outcomes that receive less attention but are clinically important deserve explicit discussion. First, bone mineral density. The caloric restriction group showed small but statistically significant decreases in bone mineral density at the hip and lumbar spine compared to controls. These decreases were proportional to the weight lost and were in the range of 1 to 2%. For a population already concerned about osteoporosis risk, this finding is relevant. The authors emphasized that the reductions were modest and within the range expected from equivalent weight loss in other contexts. Whether they are clinically significant over longer time horizons, particularly in postmenopausal women, requires longer follow-up data. Second, quality of life and psychological outcomes. A frequently cited concern about caloric restriction is that it impairs quality of life through hunger, food preoccupation, and mood effects. The CALERIE data showed no statistically significant decrease in overall quality of life scores in the caloric restriction group compared to controls. Mood scores, sexual function ratings, and sleep quality were not adversely affected at the achieved level of 12% restriction. The participants did report modest increases in hunger early in the intervention that diminished over time. This finding does not mean caloric restriction is easy or universally tolerable. It means that in the intensive support context of this trial, at 12% achieved restriction, the psychological burden was not detectably worse than the control group. Now for the critical analysis. Several limitations are important. First, the CALERIE participants were healthy, relatively lean adults recruited and screened at research centers. They were younger on average than 55. Whether the results generalize to older adults with chronic conditions, polypharmacy, or lower baseline lean mass is not established from this trial. Second, the achieved restriction was 12%, not the targeted 25%. The CALERIE trial demonstrates what is achievable with substantial institutional support in motivated research volunteers. The practical sustainability of even 12% restriction in real-world settings over years is unknown. Third, the cardiometabolic improvements observed in CALERIE are consistent with the effects of equivalent weight loss by any means, not specifically caloric restriction as a modality. The mechanisms unique to caloric restriction, beyond the weight-related effects, are an active area of research. A 2022 follow-up analysis of CALERIE data by Krauss and colleagues in the journal Aging Cell found that caloric restriction produced changes in gene expression pathways related to immune regulation and proteostasis that were not fully explained by weight loss alone. This suggests biology beyond simple weight reduction, but this molecular evidence is preliminary and mechanistic rather than clinical. A fourth point, the bone density finding is specifically worth discussing in the context of adults over 55, where baseline bone density is lower and where the clinical threshold for fracture risk is more likely to be crossed by small reductions. If you are considering any dietary strategy involving reduced energy intake, having a baseline bone density DXA scan and discussing calcium and vitamin D adequacy with your physician is relevant. This is especially true for postmenopausal women and for adults taking medications that affect bone metabolism, such as corticosteroids. 3 takeaways. First, the CALERIE trial demonstrates that sustained moderate caloric restriction, achieved without malnutrition or extreme dietary behavior, produces measurable cardiometabolic improvements in non-obese adults. The achieved reduction was 12%, roughly 200 to 300 calories per day below baseline. For context, eliminating one daily calorie-dense snack or reducing portion sizes moderately can produce this magnitude of reduction. The clinical significance of the resulting weight loss and biomarker improvements is real, even if modest at the individual level. Second, the bone density finding is a reason not to approach caloric restriction in isolation. Adequate dietary calcium and vitamin D, resistance exercise to maintain bone mechanical loading, and baseline assessment of bone density before undertaking sustained restriction are all relevant considerations for adults over 55. A registered dietitian can help ensure that any caloric reduction preserves nutritional adequacy across all essential micronutrients. Third, the gap between the CALERIE target of 25% and the achieved 12% is itself an important finding. Voluntary caloric restriction at the levels that produce dramatic effects in animal models is extraordinarily difficult for humans to sustain outside of highly controlled research settings. Recognizing that even modest, sustainable reductions have measurable effects is more useful than pursuing aggressive restriction that is unlikely to be maintained. To summarize, CALERIE was the first multi-site randomized trial of caloric restriction in healthy non-obese U.S. adults. Participants achieved a 12% reduction from baseline over 2 years and showed significant improvements in cardiometabolic biomarkers with no adverse effects on quality of life at that restriction level. Modest bone density reductions were observed and deserve monitoring. The trial did not include adults over 55, limiting direct extrapolation. This is educational content, not medical advice. Always consult your physician before changing your dietary regimen. Studies are preliminary, individual responses vary. Link to our free biomarker guide is in the description. New evidence reviews every Tuesday and Friday.